Yeah, thank you very much for the invitation and the chance to present my data or my ideas. So as you already said, I'm in now lovely Olsztyn, as you see it in the background, the Polish Academy of Science. So good morning of those who are in Europe and good afternoon for those of you who are in Korea. So we are talking about omics technologies in food and health. And since this is a data meeting, I want to emphasize the aspect of data. So data stored in the genome and in the epigenome, that is what I will focus on. So I guess you're all aware of that sequencing, basically massive parallel sequencing, is the workhorse in modern bioscience. So we have the human genome since some 20 years, but we use also the sequencing technology, of course, to sequence other species as extinct human species like Neanderthals, other primates, many other species, and now focusing further on humans, also different human populations. For example, like in the big biology project, 1,000 genomes, 26 different representatives from 26 different populations, all facing aspects of different type of diseases like cancer in the TCGA project or epigenome projects, and so on and so on. And also just to briefly mention that many techniques to study the details in cells are also used by the sequencing technology. But let's have first a moment on the genomics aspect. So I guess all of us are aware of, we have 2 copies of our genome in each of our cells, and we have many different cells, 6 trillion cells with a nucleus. So here I'm not counting in the red blood cells. So we have, kind of speaking in computer language, 6 gigabytes in each of 6 trillion cells, and it's the same. And if we compare different individuals, we have variations between us in different forms of single nucleotide variants, deletions, insertions, structure variants, and so on. But altogether, they make only 1% of the variation. So basically only 1% of our genome are different between all of us. But this 1% are those genomic or genetic impact on diseases, which I will come back to the very end. But let's first zoom into a cell. A cell looks in an electron microscope with lighter areas and darker areas. The lighter areas are the euchromatin, which in fact, although it takes here a lot of space, is only 10% of the epigenome. And the epigenome is basically expressed here. Chromatin is the physical expression of the epigenome. So we see chromatin, and this open chromatin, euchromatin, and 90% of our genome are in heterochromatin and therefore not visible and not accessible. So if we compare 2 different individuals, we have on one side variations in the sequence. This is what you see here with the capital letters. The T is the same with these 2 different individuals, but the A and G here is different. It's the same in all tissues of the individual, but different here in another individual. If I now look on 4 of altogether 400 different tissues and cell types, you see here indicated with methylation marks, each tissue is different in the epigenome. So that means we have in each person at least 400 different forms of the epigenome and only 400 different forms and only 1 genome. So that we have a far higher amount of data stored and formed of the epigenome. And that is of course also different between individuals. So how experimentally addressing the epigenome? Big biology projects like ENCODE, Roadmap, Epigenomics, Phantom 5 and others were doing that, and the epigenome has different type of layers. So it's basically like you have different storage elements. You have a DVD, you have a hard disk, you have USB sticks, and that is what we have on a different levels. A larger structure of the chromatin down to here methylation changes a different position of the genome. So these different levels of the epigenome, each of them is storing information. So one concrete example, also very close to my own research, is if we study vitamin D signaling. And maybe one thing I forgot to say here, these big biology projects, they are just describing for many different tissues the basal state. But if you want to do a real experiment, for example challenging cells with vitamin D or any other type of drugs, then you need to do the experiment yourself. And then you need to record, for example, binding of proteins, chromatin changes, chromatin accessibility, and this data needs recorded by these different type of methods mentioned here, like ChIP-seq, FAIR-seq, RNA-seq for the transcriptome, and finally are integrated by bioinformatic methods. So this integration, of course, is one or maybe the most important part of all this data handling. So we have different layers of the epigenome, as I mentioned, and these different layers, they change when we change something in the experiments, in the setting, or if we go to a different tissue. And in this way we have different layers, and these layers need to be integrated, they need to be aggregated to tell at the end a story, and that is the core of what we're doing with bioinformatics, bioinformatic handling of the information that is in the epigenome. So the link now to nutrigenomics and nutritional epigenetics. The definition of nutrigenomics is we have many molecules within our diet, and many of them, or a couple of them, are having the ability to directly activate transcription factors. Then some of these molecules get metabolized and then activate transcription factors or chromatin modifiers. Or last but not least, some of the molecules from our diet activate a single transduction cascade, the end of which are chromatin modifiers or transcription factors. So in three different ways at least, we have the ability that our nutrition, our breakfast, our lunch, our dinner is talking with our genome/epigenome. And now if I focus on nutritional epigenetics, you have to think about, we have here our epigenome represented here with nucleosomes in different states, different information status of the epigenome. Proteins handling that, basically reading devices, writing devices, like we have it on a computer. And these devices, and that's the important point, are modulated in their activity by molecules indicated here as examples, which are derived from mostly plant-derived food components. So it is in green tea, in soy, in grape, cocoa, curcuma. So in this way we have our diet or components of our diet directly talking to the epigenome and modulating the storing ability of the epigenome. So again, we have the problem of integrating the data. From my personal perspective and that of many other people, one is starting at the bottom with the genome/epigenome and records and this describes the data here. But of course, depending on your interest, you can also measure the transcriptome, proteome, metabolome, microbiome and record this data. Emphasize again, the data is important, and integrate the data to make a most comprehensive story. So now coming just in a point to the environment, so the modulator of that. So I gave you an example of different food components or vitamin D, but basically everything that is representing our life can have this effect on the epigenome. So therefore I come to the conclusion telling you my main message, and that is from the first moment of our life, so directly after fertilization to birth, 9 months later, for the whole life until the last day of our life, we are recording data in form of our epigenome in these 400 or more different cells and cell types and tissues. And this recording is basically describing, on one side, just a collection of data, but of course, since it has a functional consequence, it is modulating our risk of getting diseases, of forming our phenotype. If we are healthy, everything fine, but if we get diseased, this phenotype of being diseased is to some extent driven by our genome, but on average this is just some 20%, and the remaining 80% come from our epigenome that is modulated by the environment. And basically that's my story, and I'm happy to take any questions.
Okay. Thank you, Carsten. Now